الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. Good evening everybody. رمضان مبارك عليكم جميعا. Congratulations for the month of Ramadan. Actually, it's really my honor and pleasure on behalf of the Saudi Society of Blood and Bone Marrow Transplantation to have tonight with us one of the eminent professors and clinical scientists in the field of hematological malignancy, the well-known Professor Lucy Godley. She's a full professor at Prospera Foundation in Oncology section of Hematology Oncology at the Department of Medicine and Human Genetics, University of Chicago Comprehensive Cancer Center in Illinois, in the US. Professor Godley began her scientific and astonishing career in several well-known laboratories across Yale, Harvard, and the NIH in the US. She completed her medical training at Northwestern, then followed that by doing the internal medicine and then hematology and oncology residency fellowship at the University of Chicago, where she joined the faculty in 2013. She's well known as a physician scientist with a strong research and clinical responsibilities. Professor Godley seeks in her career to understand the disease on the molecular basis and its and why there a link between genetic mutation and hematological malignancy that able to bring the perspective on the care of her patients. Currently in her laboratory, she studied the inherited hematopoietic malignancies and how to control the differentiation potential in the cancer cells by modified cytosines. She and Dr. David Wu from the University of Washington, the co-chair of myeloid malignancy variance expert panel within ClinGen. So without any further delay, we want to welcome Professor Lucy Godley. But before that, we want to thank our sponsor for tonight, the Saudi Society for Blood the Saudi Society of Blood and Marrow Transplantation. Kindly, Professor Lucy, without any further delay, the floor is yours. Thank you.
Thank you all so much. It's really an honor for me to be invited from my colleagues so far away. And I hope one day that I can come in person to see all of you. It's really an honor during your holy month of Ramadan and so late at night for all of you to take time to listen to what I think is very important, the diagnostic approach to bone marrow failure disorders. I'd like to begin by acknowledgements. And first to my patients, you see this on the left. This is a thank you note that my the patient's daughter gave to me for taking care of her mother. But really, I thank my patients because I was in the lab working on cytosine modifications, doing very basic science. But when I was caring for my patients in the clinic, I took their personal and family histories and really felt like they were programmed to get these diseases. And I started diagnosing these individuals and families with inherited predisposition. And I started to think that I was taught these syndromes were extremely rare, but I was thinking it cannot be so rare if I'm diagnosing them all the time. And so I really think that as a field, we need to appreciate the germline predisposition that many of our patients have. And in the Middle East, I think that you probably have a different spectrum of those inherited predisposition because you have a different patient population than what I see in Chicago. But the same phenomenon is true, and I hope to motivate you in that way. In my lab, we have a lot of individuals who are really very dedicated to this subject. And I really want to thank my colleagues in the genetic services lab, especially Soma Das. When I had this feeling like germline predisposition was more common than what I was taught, we needed a diagnostic platform to test the patients. And Soma was able to develop a comprehensive platform that I'll talk to you about. And this has been very important for diagnosing our patients. In our section of pathology, Jeremy Siegel runs the molecular profiling that we do on the leukemia cells, and they share that information with us across all the patients with hematopoietic malignancies and bone marrow failure. And we look at the data and we surmise who is at high risk for having that allele as a germline mutation, because of course, the germline mutations are in every cell, even the malignant cells. So when you see a RUNX1 mutation, a GATA2 mutation, et cetera, how do you know that that is somatic or germline unless you test? And so they're very open with us. We share this information and we identify these individuals in that way. Dr. Vardaman was on the World Health Organization committee that considered revisions to the diagnostic criteria for leukemia, and he was a big advocate for including germline predisposition to myeloid malignancies as a provisional category in the World Health Organization. And I would say that I tried very hard to get the WHO to make that category germline predisposition to hematopoietic malignancies, not specifically to myeloid, because I actually think the lymphoid malignancies are more genetically determined than the myeloid. It's just at the time we had more papers on myeloid predisposition, and that's why the diagnostic category is skewed to myeloid. But really all of the hematopoietic malignancies have a strong germline component. And if we have more time at the end, we can talk about that. So I think of patients on multiple levels, as I'm sure many of you do. People come with their baseline genetics, their baseline epigenetics, and then over time, they're acquiring genetic changes in their hematopoietic stem cell or in their tumor initiating cell. Many of these we know for myeloid malignancies are within epigenetic modifiers like DNMT3A, ASXL1, and TET2. And these are bringing epigenetic changes into the hematopoietic stem cell. And I am a firm believer that when we get to the point that we're really molecularly defining all of these levels for all of our patients, then we're really going to be able to develop effective strategies, maybe even to prevent leukemias in the people who start with a genetic risk. So let's focus today on the genetic risk, especially with bone marrow failure. But just to make the point, and I have a couple slides on this, people always say, oh, these syndromes are so rare. First of all, I don't think they're that rare. So I think if we open our minds to the possibility that they're not so rare, we'll start diagnosing them. Here on the left, you see the current genes that are known to predispose to myeloid malignancies only. In the next column, we have lymphoid malignancies or immunodeficiencies. And again, I have a bias that for many of these germline predisposition to immunodeficiency, many of them will be connected eventually to lymphoid malignancies. In the third column here, we have risk for both or either myeloid or lymphoid malignancies. And on the far right, you have either hematopoietic or solid tumors. And here you can see that some of these are really umbrella terms. So I'm trying to think of different ways kind of to make my point. And here in the United States, we have sort of a riddle. How much does a ton of feathers weigh? And of course, the answer is a ton. It's heavy, right? Each feather is very light, but in conglomerates, they're very heavy. Likewise, each syndrome may be somewhat rare, but there are so many of them that this is common as a phenomenon. And if we just think about bone marrow failure and, for example, dyskeratosis, just from bone marrow failure, dyskeratosis congenital, look at all the genes we have to think about. So these big feathers at the bottom are umbrella terms. There are many genes within here. And so when we think about bone marrow failure, we have to do a lot of searching through the germline to find some bone marrow failure is germline determined, some bone marrow failure is acquired. And the only way to know is to test them. And today I'm going to focus on the germline just because that's really what I study. And I think it's underappreciated. So we did a study that's just been published a couple months ago. So I wanted to share that result with you. Several years ago, you know, people are always saying, well, this is so rare. This is so rare. So I'm trying to think, how can we show people it's not so rare? What patient population should we study to actually measure the frequency of germline predisposition? we decided to take the population of young people with myelodysplastic syndrome or aplastic anemia, bone marrow failure. Now, there's a diagnostic overlap between myelodysplastic syndrome and aplastic anemia, as you know, and that's why we took patients with both of those diagnoses across that spectrum. Here you can see this is data from the United States. I assume in Saudi Arabia, it's very similar. If you're young, it's very rare to have these diseases. These diseases occur in more older populations. And so if we take people, and this was arbitrary, between the ages of 18 and 40 years old with myelodysplastic syndrome or aplastic anemia, how often do they have a germline mutation? Now, unbeknownst to us, the pediatricians were doing the same kind of study from their population, which goes from 0 to say 25, 30 years old. So what tissue did we use for the germline? Unfortunately, for most of these individuals, the only sample that we had was a bone marrow aspirate. It was the diagnostic bone marrow biopsy that defined this diagnosis. And we cannot make DNA just from the bone marrow aspirate because mostly we're going to be looking at hematopoietic cells. So for the vast majority of these samples, we had to culture the non-hematopoietic mesenchymal stromal cells. These cells are multipotent and give rise to the adipocytes, the chondrocytes, the osteocytes within the bone marrow. They're part of the bone marrow niche and they're non-hematopoietic. And we define germline to be that the variant or the mutation was present in the mesenchymal stromal cells as well as the hematopoietic cells. So from two different lineages, we see the variant then is we define to be germline. For some of these individuals, we were able to culture skin fibroblasts so that we define that to be germline. And for very, very few of the samples, we use saliva, which I consider to be blood, is so contaminated with lymphocytes and other hematopoietic cells. Paraffin-embedded tissue has a lot of blood in it, and obviously T cells are hematopoietic. So I think we only did one of each of these. A vast majority, we either cultured skin fibroblasts or cultured MSCs with the variant also present in hematopoietic cells. So we took patients with myelodysplastic syndrome, therapy-related MDS, secondary AML that had MDS changes, aplastic anemia, again, very arbitrarily from 18 to 40. And we were able to collect 121 samples. You can see all of the individuals across the world who contributed samples because obviously it's rare. And here's the answer. The frequency of MDS is about 20%. And in aplastic anemia, 15%. This is very, very common. I think that if you asked most clinicians in the United States prior to this study, you have a patient who's 25 years old with MDS or patient 35 years old with MDS, how likely is this to be germline? They would not tell you 1 out of 5. So this is very, very important. Now, look at what the genes are by age. If you present, we're taking the data here from our pediatrician colleagues. If we take presentation of very, very young individuals, 0 to 5 years old, most commonly they have a germline mutation in SAM-D9 or D9L. If the individual is a teenager or young adult, 13, say to 25 years old, most often the germline variant is in GATA2. And then from 18 to 40, our data is DNA repair and telomere biology. We know that elderly patients have germline mutations in DDX41. These people present with myeloid malignancies at the same age as de novo MDS. So if you have a 75-year-old person with MDS or AML, we cannot assume that the germline does not play a role. And I think this bias that we've all been taught, if you have a germline mutation, you will present young. It's true. If you present very young for a diagnosis, there's a reason. But it doesn't mean that if you present at an average age, you don't have a germline predisposition. And before we got on the webinar, I was saying that I've been to Amman, Jordan twice because we have a Jordanian American family with a very particular DDX41 mutation that I feel is probably a founder mutation within the Middle Eastern population. We can talk about that at the end. Since I've been there, the doctors at the King Hussein Cancer Center have identified that mutation in another family. So I think it's quite possible. DDX41 is the most common germline predisposition syndrome we see in Chicago, and there are very particular mutations in particular populations. And you have a very different population from what I see. You will see unique germline mutations and unique genes for your population. Now we have a knowledge gap from 40 to 80 years old, and I would love to fill this gap. So I've been trying to write grants to do the study in the whole age range, because I think age of presentation is merely a surrogate for the biological pathway. If you present very young, you are likely to have a mutation in these genes. If you present during young adulthood, a different set of genes. If you present in the older, a different set of genes. And we don't have the data across the entire age range, but that is what we need to get. Hopefully we can do it. So what is our current criteria? When we see a patient, how do we decide to do the germline predisposition? I would love to tell you that we do this in every single person walking in the door. I hope soon we will come to that, but right now we have certain criteria. Number one, multiple cancers in a single individual. We have this idea that therapy-related leukemia is a leukemia that comes after usually a primary tumor or maybe autoimmune disease for which chemotherapy has been given, or a rheumatologic condition for which chemotherapy has been given. And there's been a bias, especially at University of Chicago, that it's that chemotherapy or that immunosuppression that is causing the leukemia. I think it's both. There's germline predisposition, and then there's environmental insults from radiation and chemicals. And that's what's giving you the predisposition to therapy-related disease. So we also have people we call double cancer. These are people who first have a hematopoietic malignancy, survive long enough to get the solid tumor. It doesn't matter whether they have the solid tumor first or second. The frequency of positive testing in someone with 2 or more cancers is 20%. This is just being published now in leukemia as well. This is very, very high. So again, if someone has multiple cancers, they are reading out for you their germline predisposition. We talked about this already. If you're diagnosed with a hematopoietic malignancy well outside the normal age range, young MDS, for example, aplastic anemia in the young, we need to think about the germline. But again, I'll say this again, if you present in an average age for that diagnosis, it does not mean that the germline is not contributing. And I think that we've been sort of taught the wrong thing, or we've taken this corollary incorrectly. If we have someone with a heme malignancy and they have one other heme malignancy or young onset solid tumor, say a mother with breast cancer at 45, we go ahead and test that individual. Or other heme abnormalities in the family, other cytopenias, bleeding, or other anemias. This can be harder for people to know about within their families, but we try to always ask. So we take this personal and family history very carefully. And what has made the difference for us is that we have a person, he's a college graduate, he's not a genetic counselor, he's not a nurse, he's not a doctor, but he's trained in this, and he goes to every single bone marrow biopsy, outpatient or inpatient. He takes the personal and family history, and he also looks at the molecular profiling that was done in the leukemia cells. And we decide if the people meet these certain criteria, if they have high variance allele frequency. High for us is over 30% of any gene that we know confers germline susceptibility. We go ahead and do a skin biopsy right at the time of the bone marrow biopsy. We're already cutting into the skin. We just get a skin sample, grow the skin fibroblasts. We also try to do this at the time of a matched related or even unmatched or mismatched related transplant. And I can give you some horror stories, and I give you a little bit of a scary horror story at the end, since this is a transplant group. By all these different ways, we find these patients, we try to do the skin biopsy, again, at the time of the bone marrow biopsy. Occasionally, I do just a separate skin biopsy, and we grow the skin fibroblasts. We culture the skin fibroblasts. Now, we'll come back to this because in the case of bone marrow failure, this can be a little difficult, and we'll talk about that at the very end. We make DNA from those skin fibroblasts, and right now we're doing an augmented whole exome. What I mean by augmented is we have a whole exome platform, and we add in primers that bring down non-coding regions. The GATA2 enhancer, the ANCO D26 promoter, the DKC1 promoter. These are not typically covered well by an exome, so we pull them down. And then we look for single nucleotide variants or copy number variants in relevant genes. If it's positive, we do very extensive cascade testing. As I said, I've been to Amman. I've met with the family, the people in Jordan, who are the relatives of my family here in Chicago. So I go to very far lengths to do this cascade testing. And if it's negative, we do research and we discover new syndromes. And I'm sure in your population, you will find new syndromes if you are looking like this. Now, these data come from our old version of the clinical testing, which was a next generation sequencing panel with a microarray to find the copy number variants. And I just want to emphasize how common germline copy number variants are. I was naive when we started this testing. I did not realize that germline copy number variants were going to be so common. And I'm very thankful for the laboratory for insisting on doing a paired microarray, just in case it turned out they were absolutely correct. We would never have found these without that. What's the diagnostic answer? You using the criteria that I gave you before, you see we test across the entire age range of life, and the answer is 20%. We keep getting 20, 20, 20%. This is very, very high. 15% in kids, 20% in adults, very high. And from genetics standpoint, is too high. It means our criteria are too strict because we want a diagnostic rate closer to 5%. So this means we are not even testing enough people. So let's talk about this practical aspects for bone marrow failure, because I was asked to speak specifically to bone marrow failure. We know that the skin fibroblasts do not grow well for people from bone marrow failure. So if we have a patient with aplastic anemia or bone marrow failure, we often take 2 skin punches. And from one punch, we try to culture the skin fibroblasts. And from the other punch, we make DNA directly. This one is not so great because there is blood in the skin punch. And we make the DNA, we have some contribution from the blood, but it's better than nothing. And then we test the germline status, occasionally only from this sample because our skin fibroblast culture will fail. Now, we define germline in one of 2 ways. Number 1, it's present in cultured skin fibroblasts. So if we get the answer from this DNA sample that has blood contamination, we use the second means to define germline, which is we see that variant in a relative. So we do cascade testing. As soon as we see that variant in a relative, we know that it's germline. So some people say to me, I have somebody with bone marrow failure. I'm going to do a bone marrow biopsy. I'm going to do molecular profiling. Why can't I just use the molecular profiling to figure out who's at high risk for germline? And there are several reasons why we should not use somatic testing. We should do proper germline testing. So when I say somatic, I'm saying the DNA comes from malignant cells or from, say, aplastic marrow, as opposed to proper germline. So number 1, I would say somatic mutations, we know what they look like, right? We've done so much sequencing now that we know what genes are the genes to look for. That means the platform is relatively stable, but it often does not include the genes that confer germline risk. So I told you that for us, DDX41 is the most common inherited syndrome. It is not present in most commercial laboratory panels in the United States labeled as inherited myeloid malignancy. To me, this is malpractice that the companies would market a test that misses the number 1 gene, right? And these are very reputable companies. And I'm pushing them and pushing them to do the right thing, which is expand the panel so that it's accurate. Now, for the germline, we are still discovering more syndromes, right? So this means the platform has to expand over time. And this is why the laboratory went from a panel-based test now to augmented whole exome. So they can add genes anytime they want. They have the data from the exome. This is probably the most important reason biologically. Hematopoietic tissues undergo somatic reversion easily. So if you don't see anything in DNA that comes from either blood or bone marrow, it can be a false negative very commonly. This is why SAMD9 and D9L were missed in the pediatric population for decades, because people were taking children with monosomy 7 and they kept sequencing blood and bone marrow. Finally, someone sequenced the skin and found the germline variant. So we know that somatic mutations are pretty much always in the exons. So you can define these next generation sequencing panels very easily. You take the commonly mutated genes, you panel the exons, you're done. And you can use hematopoietic tissue. That's the affected tissue. But when we're talking about the germline, we can't do that. And so we have to use true germline sample, which is a pain in the neck because you have to grow the skin fibroblast, and then you have to use a panel that's comprehensive. Now, if we look at particular alleles, most of the time, we cannot say if it's somatic or germline. The same allele in RUNX1, say an R204Q, can be somatic or germline. So you cannot look at the variant from leukemia cells and tell. The variant allele frequency is a hint, but it absolutely does not tell you. There are two genes that we see here in Chicago that we can look at the allele and pretty much know if it's germline. That's CHEK2. We have a very large population of individuals here who come from Poland. We're the second largest Polish city in the world. And so it's not surprising that we would see CHEK2 because of the Polish founder mutations. And DDX41. For us, again, the most common germline predisposition syndrome with particular variants from certain populations. The M1 question mark, the D140 frame shift in Caucasians or Northern Europeans, the A500 frame shift from Japanese, and I think the P78 from the Middle East, but this is still to be determined. Now, I just want to end in the last couple minutes with a little bit of a scary scenario because all of you are transplanters, and transplant makes me very nervous from lots of perspectives. This is a true story. I was talking about germline predisposition and going on and on and on about DDX41 to my transplant team several months ago. And when I finished my talk, one of the nurses raised her hand. She said, "Lucy, I think we have one of those DDX41s." And she was absolutely correct. They had a patient with leukemia. The leukemia cells were profiled on the outside, and she had looked at the molecular report, and she saw the D140 frame shift at a high variant allele frequency. This is, again, the Northern European common mutation. And then she sees the R525H at a lower variant allele frequency. This is a hotspot, acquired mutation, often in the context of a deleterious germline variant. So I was saying to them, if you have patients coming for transplant who have a DDX41 at a high variant allele frequency, especially with an R525H at a lower variant allele frequency, think about the germline. And she raised her hand. She said, "Lucy, I think we have one." And they absolutely did. Now, they put their patient in remission. Now we think about the donor. We prefer related donors. What if we use a related donor without thinking about the germline? We are using hematopoietic stem cells, potentially that have a mutation in them. We think about RUNX1, GATA2, TP53. Those are master transcription factors. And cells need these transcription factors, especially hematopoietic stem cells. They need RUNX1, GATA2, CEBP alpha, et cetera. They don't function normally. And so it is a disaster if you use these as a donor. We have seen poor mobilization, poor engraftment, poor graft function, donor-derived leukemias in the recipients, leukemias in the donors. We're giving them GCSF typically to mobilize them. It is an absolute no to use hematopoietic stem cells with germline RUNX1 or CEBP alpha mutations. Full stop. This has been seen many, many times, published in the literature. Absolutely no. It's much harder to know what to do with hematopoietic stem cells that have a BRCA1 mutation or a BRCA2 mutation. Are those okay to use? Makes me very nervous. I can tell you that we had this scenario several months ago with a related donor who had a CHEK2 mutation. I told you CHEK2 is very common. We have a very Polish bias in our population in Chicago. And I warned the transplant program, I said, these stem cells are not normal. I cannot prove to you that you're going to have a problem, but CHEK2 is in homologous recombination. It's a major DNA repair pathway. Be very careful. We had graft failure. Now, can I tell you that it was graft failure because of the CHEK2 mutation? I cannot prove it, but I'm very worried. And so I would say be very, very careful at the time of using the relative. What happened in this case? Well, we knew about DDX41. We knew this was germline in the individual. So this team went with the unrelated donor. What happened? We did engraftment at day 30. This individual was all donor, but they went ahead and did molecular profiling. Why? Because they wanted to show that the DDX41, which was from the leukemia, was no longer in this individual. But we're 100% donor. You are looking at the DNA from the donor, right? So these tests should be done in parallel, not in series. If you're 100% donor, I think the pathologist should only be reporting on the leukemia-specific mutation. You should not be revealing the germline genetics potentially of the donor. And that's exactly what happened. We see the CHEK2 I200T at a variant allele frequency of about 50%. This is from the donor. So this donor has a germline deleterious variant in CHEK2. We've changed from germline DDX41. Now we have a germline CHEK2. And we've put these stem cells into a replication stress by asking them to divide and fill out this new bone marrow. Hematopoietic stem cells don't like to divide. They're quiescent cells. So we force them to go through DNA division. They have mistakes, and now they have a major defect in their ability to repair that DNA using homologous recombination. They're going to use backup DNA repair pathways that are error prone. So this individual is now at risk for a donor-derived leukemia, and we've seen it. So I'm very scared by transplant. Transplant is ethically compromised. We're asking our relatives to have genetic testing that they might not otherwise have. I can talk about scenarios where that's been true. And we're revealing germline DNA information to the recipients without the permission of the donor. At least that's what's happening in the United States as I see it. And so I would really like to see changes in our transplant program, and I think throughout the world. With that, I just want to remind you that these syndromes are really common in my mind. They might individually be somewhat rare, but there are so many of them. I think we need to think about this. And I think I've taken about a little bit more than half an hour. So maybe I stop sharing at this point, and I'm very happy to take questions.
Wonderful and well-conducted talk, Professor. I really, or we really enjoyed it fully. Believe me, before your talk, I wasn't that convinced about the impact of the germline upon myeloid neoplasm, but you fully convinced me about it's really common, and we should look carefully when we diagnose those patients in advance to do full sequencing, especially in our community. I'm wondering if Dr. Ahmed Al-Askar want to have any remarks about this talk.
Sure. Thank you very much, Lucy. Excellent, very informative talk, as pointed out by Dr. Osama as well. Very interesting, actually. And as you said, we as hematologists, especially the adult, I guess, are less exposed to these, especially the germline and mutations and how to interpret this. And that's definitely an eye-opener, I guess, for many of us. We see actually lots of cases, I would say, in Saudi Arabia, that I think is inherited. And actually, sometimes we face difficulty finding a donor among the siblings. And we have a number of cases as such. So that would be something that we could even explore and work together on and exploring this. I'm sure we have new syndromes, and we actually explored some, and we do have new syndromes.
Yeah, I'm sure it's true. And also there are cultural differences. In the United States, of course, it's a melting pot from people all over the world. So we see some people who really, really do not want to know. They don't see this information as being empowering. Whereas of course, I hope that the information is very empowering to people, because if they know that they don't have the family mutation, they don't have to worry. And if they do, we can put better surveillance into place. But when I was in Jordan, the doctors there were explaining to me that some families, at least there, they feel like finding out something like this is like a stain on the family or something like that. And so I think there are different cultural, cultural aspects that each, country has to think about, and what's the appropriate way to address this with their patient population.
Yes, absolutely. We found it actually not as stigmatizing as been mentioned, but I'm sure many families will be interested to explore this, especially when they have a sick ones.
Exactly. Yeah. So I think we have a couple of questions. The first one, what do you think if we use peripheral blood in remission? Or you could even say bone marrow biopsy in remission. The problem with using the hematopoietic cells is this issue of the somatic reversion. So if you see a mutation at remission, you certainly know that it's there, and you have a high suspicion that it could be the germline. But if you don't see anything, there still could be a germline mutation. So it's just not as sensitive to use it. That's why we prefer the skin biopsy. And saliva is blood for me, because it has so many lymphocytes, especially, and other blood cells. So even fingernails have monocytes in them. So true germline is cultured skin fibroblasts or DNA from hair bulbs. It's just that most of our patients have leukemia. They have they've had chemotherapy, they don't have their hair, but they might have eyebrows, but they don't want the hair plucked. Our patients don't want it because it hurts, and they don't want to lose any more hair. So we have tried, but our patients don't want the hair plucked. And also you don't get much DNA from a hair bulb. And so you can't do a lot of the assays that I'm talking about. The skin fibroblasts, when they grow, is almost a renewable source of DNA. They will senesce, but we can use them scientifically, for instance, to make induced pluripotent stem cells. Then this is very useful for science. So we always ask our patients to participate in our research, and then it's very useful. The next question is, if culturing of the skin fibroblasts is difficult or not available, what's the alternative? So certainly if you're not able to do it, then you may have to use the buccal swab or the hematopoietic cells. But I would say we can do it. First of all, we're happy to send you the protocol for making the skin fibroblast. And if you have the amniocentesis procedure, I don't know, I assume it's done in Saudi Arabia. The lab that does the amniocentesis is the constitutional cytogenetics laboratories. They're used to growing fibroblasts. Those are the cells that they're growing. And so when we started this, we actually asked the cancer cytogenetics lab to try to grow the skin and the constitutional lab. We kind of put them in competition. And the constitutional lab was better because that's what they're used to doing. And so if you have that procedure, then you almost certainly have the technology to do this. The other thing is that we have grown the skin as far away as Israel. So we had a patient in Israel who wanted this kind of testing. We sent the skin media all the way to Jerusalem. The skin biopsy was done. It was sent all the way back to Chicago, and we had the skin grow. So I think it's better if you do the culture there, but if it's not possible, we could try to work something out, where we send the samples. Let's see, considering that some germline mutations in familial leukemia can display reduced penetrance, should prophylactic transplant be offered to those with symptomatic manifestations that have not developed full-blown malignancy? This is such an excellent question. And some of you who were at the original workshop on inherited heme malignancies at American Society of Hematology meeting, now it's 5+ years ago, will recall this was our first ethical question. When do we feel comfortable as a group doing, a lot of people use the term now, preemptive stem cell transplant? The one syndrome that I would say, very good argument, is germline CEBP alpha mutation, if that mutation is at the 5' end of CEBP alpha, because the penetrance is close to 100%. So if you had a child with a 5' end CEBP alpha mutation, your child is going to get a myeloid malignancy. And we actually had a family present at St. Jude where the family had read the literature. They were pushing the doctors to do a preemptive transplant. The doctors were nervous, not the family. And we see this again and again in the United States. The family wants to know what's going on, and they push the doctors. And so now the St. Jude has published an opinion piece in the Journal of Clinical Oncology saying that in the balance, there are some conditions for which we can argue for preemptive transplant. Traditionally, the transplanters in the United States have been nervous to do it because of the lifelong risk of graft versus host disease. If you have something like germline BRCA mutation and you're going to do bilateral mastectomy, you have a one-time surgical risk, right? The surgery is done once and you're finished. You don't have the risk going on and on. But with transplant, you have the long-term risk of GVH. So that's always been the argument not to do it. But when you have a highly penetrant disease, I think you can make this argument. Now, what we're trying to do on a research basis is understand the molecular progression. If you have a germline RUNX1 or germline GATA2, how do you get to leukemia? And we've been able to take, because now we've done so much cascade testing, we know the people who have the mutation who are decades away from earlier, from getting their leukemias. Do they have clonal hematopoiesis, for example? Germline RUNX1, germline GATA2, they have clonal hematopoiesis. Germline DDX41, no, at least not in the typical genes. Now, the other issue there, the typical age of leukemia is much younger in a germline RUNX1 and GATA2. So the question I have now is the presence of clonal hematopoiesis a molecular clock? If you have clonal hematopoiesis, are you 15, 20, 25 years away from getting your leukemia? And we won't see this in the germline DDX41s until they're 50 years old. We just have to follow people. The problem is we have to follow them for decades. And so here in the United States, we've been trying to center these patients at the National Institutes of Health in Bethesda, where they can have the funding over a very, very long period of time and help us answer this question. If we knew that B-core mutations predicted leukemia in germline RUNX1, then we could say, if you see clonal hematopoiesis with a B-core mutation in a germline RUNX1, go ahead and do the preemptive transplant. These are the studies that we're trying to do right now. We don't have the answers right now. If we think inflammation is required for that progression, can we intercept, slow the progression with anti-inflammatories? These are some of the other ideas that we have now. There's nothing definitive yet, but this is kind of what we're going to try to do. So do you recommend with aplastic anemia with a detected mutation and the identified related donor is negative, studying from the blood in the relative, should we do a skin biopsy in the relative? This is an excellent question and was posed to me by one of our genetic testing companies. Because if we're saying that somatic reversion happens in hematopoietic tissues, are we satisfied with a negative blood result from the relative? In theory, we should not accept it, right? But at some point we have to, I would say, be practical. Is it fair to ask that donor to have a skin biopsy? I can tell you right now at University of Chicago, we are not doing that. We recognize that's not a perfect thinking. And so maybe the future is going to be doing exactly that. I would say we're already failing at being very systematic at the time of transplant, as I've indicated to you. And so we're not quite at that question, but the person is asking a very, very insightful question.
Yeah.
Yeah, I think that I can still see maybe a couple more. We have time? Yeah, we have time.
Yes. Yes.
Excellent presentation. We screen sibling donors of patients with CLL who are candidates for allotransplant, since familial CLL is not uncommon. But don't you think that screening donors in general is practically impossible? You know, this is a good question. I said at the beginning, I am feeling like if the diagnostic rate is 20% in all MDS across the ages, if it's 15% in aplastic anemia across the ages, no, it's not impossible and we should do it. That's too high. The problem is we don't have the data right now. I would say for MDS and aplastic anemia, if you have diagnosed someone under the age of 40, you have the literature on your side now. You are compelled to look in the germline. You are going to get an answer 15 to 20% of the time positive, at least in our population. Now, again, in Saudi Arabia, you have a different population. It might be different in your population, but I think you should do the study and look. Will we get to the point of doing all heme malignancies? We are not there yet. It's going to take this kind of study in all of the diseases. Now, CLL, we have not done systematically, but I can tell you at University of Chicago, we have done multiple myeloma. So if the proband has multiple myeloma and fulfills those criteria that I told you, the testing positive rate is 30%. It is the highest that we see of any primary diagnosis. What are the genes? BRCA2, CHEK2, ATM, the homologous recombination, interestingly, not BRCA1. It will be fascinating for you to do a similar study with such a different patient population. We don't have a lot of people in Chicago. We have, I mean, some, but I would say we're more a Polish city than we are a city with descendants from the Middle East. So you could have a very different spectrum because it all depends on what your population is. Another question, do you have skin fibroblast registry to determine the frequency, frequency of abnormal mutations like the HLA registry? This is an excellent question. We do not have that. I can tell you skin fibroblast culture is not perfect either. We do occasionally see lower level variance allele frequencies suggesting either mosaic state or culture artifact. And that's why, especially if you're starting with bone marrow failure, I should have mentioned this when I gave you the slide of culturing the skin fibroblasts. We only let the lab culture the skin fibroblasts for 2 weeks in those individuals because we know the skin will not grow very well. And then we always do the second skin punch where we make DNA directly. So for bone marrow failure, we really limit the culture time. The longer the cells sit in culture, the more artifact you're going to see. And if we see one of those lower variance allele frequencies, then we try to get a second skin biopsy. This is not a perfect test either, but nothing is perfect. So I think the skin is sort of the best that we can do. Do I think that comparative genomic hybridization will be helpful in defining significance of the abnormal mutation? Yes. So when I was talking about the original version of our clinical pipeline, the array that I was talking about was a comparative genomic hybridization array. It's extremely important. Again, in our patient population, we see a lot of germline copy number variants. So the testing platform has to be comprehensive. We see a lot of germline deletions in RUNX1, in GATA2. We had never seen it in DDX41 until recently. And so we now have what to my knowledge, is the first family with a germline DDX41 deletion. In our patient population, it is much less frequent than what we see in RUNX1 and GATA2, but it's there. And so unless you look, you will miss it. High variant allele frequency, is it predictive of germline? It can be. So, the variant allele frequency, I would say it's very dependent on the cytogenetics of your, if you're getting this from the leukemia. If you're looking in the leukemia, say, for example, TP53, you can have loss of heterozygosity and see a very high variant allele frequency just from somatic. And TP53 is one of the ones where the exact same allele can be germline or somatic. So if you see a high variant allele frequency, we prioritize that patient for proper germline testing, which again for us is, if we can, culture skin fibroblasts. What about young patients with solid cancer? Should we ask about skin fibroblasts? Again, a great question. You probably saw that paper that just came out on lung cancer. We always think of lung cancer, at least in the United States, as being smoking related, but of course, most people who smoke don't get lung cancer. And so this was a study of, I believe it was small cell lung cancer, looking at the germline. Guess what the answer was? 20% have mutations in DNA repair. So again, it's the germline and the environment. Look at mesothelioma in the United States. This is, thought to be asbestos exposure. But a lot of the people who get a mesothelioma have had very minimal asbestos exposure, or you can't even find the asbestos exposure. Well, guess what? 20% of those people have a germline mutation. So I don't think there's anything special about any cancer. Cancers are cells that divide too much, and that all cells need DNA repair. And so they need DNA repair, they need telomeres. So again and again, you see all the same genes coming up. Maybe the future is going to be anybody diagnosed with cancer is going to get the germline, or maybe at birth, we're going to do people's germlines. I mean, who knows what the future is going to bring. Right now, though, I would say at University of Chicago, we are using the criteria that I shared with you on that slide. And I'm happy to share my slides with you if you would like them. That's our current practice, but I would say we keep changing what we're doing. Every few months, we do something slightly differently. And my hope is that within a few years, we will be testing every patient with a heme malignancy who comes in for the germline, because however we look at them, whether it's double cancers, therapy related, positive family history, mutations seen in the leukemia cells, multiple myeloma, MDS, whatever it is, it keeps being too high of a positive finding. And so we really should be testing a lot more patients. Somatic reversion. So I'm sorry, I did use that term, making an assumption. So somatic reversion means that the cell fixes itself. And so if the cell detects a mutation, it fixes itself from the other allele. I didn't make the point that, and I should have, most of the syndromes that I'm talking about are autosomal dominant. So we have 2 copies of every gene. 1 copy is bad, 1 copy is good. The cell is able to take the good copy and transfer it over and replace the bad copy. And hematopoietic cells are very good at it. And so hematopoietic cells fix themselves very easily. And so if you're using hematopoietic cells, they may have fixed the problem. And so you'll miss the germline mutation. And this is how SAM-D9 and D9L were missed for decades, because people weren't studying the skin. The skin is much less efficient about fixing itself. And so that's just a caution. And when we've looked in the literature, basically every single gene that we're talking about here, there's an example of somatic reversion in hematopoietic tissue. So it's not surprising, right? The hematopoietic stem cell is, I mean, the bone marrow is the most proliferative organ in the body. So those cells need to fix themselves. They need to be very good at homologous recombination. They need to be very good at fixing mistakes, and they are. But that's a problem for finding the germline mutations in those cells. If we're going to test a group of patients with heme malignancies, would you recommend using targeted or just go to whole exome or whole genome? Yes, whole genome would be the best. In the United States, right now, it's too expensive. So we do the poor man version, which is the augmented whole exome. We take an exome, we spike in those extra probes. You probably saw the New England Journal paper of Washington University. They're doing whole genome on leukemias, right? To define the cytogenetics, define the somatic. Will that be the future? Just genome everybody? You know, genome the skin fibroblast DNA? It might be. Right now, it's prohibitively expensive, so we can't do it. I was saying before we started the webinar, in the United States, the augmented whole exome costs $200 US. The whole genome, $1,000. So I can do 5 exomes for 1 genome. But these numbers are changing. And, you know, I don't know what the cost is there, but if you're able to afford whole genome, yes, it would be better, because I'm sure there are other enhancer promoter variants that are causing these syndromes that we are missing by doing an augmented whole exome. In the chat, do we have, let's see, can any somatic cells from other body organs other than skin be useful? 100%. You could take lots of almost any other cell. You see, the other problem, I didn't make this point, but the other problem with the blood for doing germline, the blood is a very unusual organ, right? Because the hematopoietic stem cells put their output into the blood which circulates. There is no other organ where the stem cells of that organ put their progeny everywhere. Think about the crypt in the colon. The colon stem cell just makes the cells for that crypt, right? The blood is unusual. It is the pooled output of all the hematopoietic stem cells. So that's why we find clonal hematopoiesis from the blood, right? We can surmise the status of the hematopoietic stem cell by looking at the blood. So this is why the blood should really never be used for any germline testing. Now we're finding all these people with mosaic P53 mutations that are found from the lung cancer colleagues, right? They're not mosaic. I mean, they are mosaics for P53, but it's clonal hematopoiesis. It's somatic mutation of a stem cell where we can detect it out in the blood. So the blood is very unusual. The reason that we started using skin fibroblasts was very practical. We do bone marrow biopsies all the time. We're cutting into the skin. My colleagues in the constitutional cytogenetics lab know how to grow skin cells. They already have the media. They already have the expertise. They did it all the time. They know how to grow fibroblasts. Fibroblasts grow in the lab. What other cells are you going to get to grow easily, right? And they're just very useful for science, right? You can program the skin to make iPS cells, which turns out to be very useful down the road. So it was a very practical choice. And I would say that has borne out over the years that it's just very easy because our patients are always having bone marrows. So we preferentially wait for the bone marrow biopsy rather than doing the separate skin biopsy, just because it's easier for people. So I think that might be at least all the questions that I see printed there.
Yeah, lovely
Yeah.
and wonderful as usual, Professor Lucy. I'm wondering, Dr. Ahmed, if you want to give a final remark for tonight's webinar.
Well, thank you, Sam, and thank you, Lucy. I mean, grateful, and you went over the questions yourself, greatly and elegantly addressed those questions. And I'm sure that if it's not for the time, we would have triggered more questions and more inquiries about this interesting topic. And Osama, because of the time, you could carry on if you have any conclusion remark as well.
Okay.
Thank you, Lucy.
Great. So as I mentioned that we were so privileged to have Professor Godley tonight with us. I stated that as hematopathologist, I wouldn't know that the impact of those genetic alteration resulting in having around a fifth of the cases being germline predisposition. And not only that, Professor Godley mentioned even for lymphoid neoplasm, it could have the case. So maybe in the future updated version of WHO, maybe there will be a provisional entity in lymphoid neoplasm. We also indicate about a new technique, at least it's not well established across clinical lab, by using fibroblast to determine the germline predisposition entities. Thank you so much again, Professor, for being with us. And we will extend our invitation for next year to have it physically in the Kingdom of Saudi Arabia. Finally,
thank you so much. It's really an honor for me to speak with you. And also, I wanted to say, if people have questions, they can email me. Noor has my email address, so go ahead and email. And I thank you so much. And yeah, I hope that we can see each other in person.
Lovely. It sounds great. And finally, as we can see, the reaction from the participants was highly appreciated. Thank you all for your attendance. And for the Saudi Society of Blood and Marrow Transplantation, my massive thanks for arranging all the necessity to have this tonight webinar. And final announcement, next week, the same time on Saturday from 10 to 11 p.m. we will have a webinar titled with Management of AML Practical Cases in Saudi Arabia by three eminent doctors in the field of adult hematology, Dr. Mohsen Zahrani, Dr. Marwan Shaheen, and Dr. Salim Dada. Thank you all. Enjoy your evening. Goodbye.
Thank you. Bye-bye.
Thank you. Bye.
Bye.